Thank you for joining us for today's presentation sponsored by GENEWIZ. Towards high-dimensional sequencing from single-cell to spatial omics. The presentation will be split into two sections, featuring our guest speaker, Dr. Yang from the Fox Chase Cancer Centre, followed by Dr. O'Hara, NGS Technical Application Specialist at GENEWIZ. Our first presenter, Dr. Yang, is an Associate Professor at the Fox Chase Cancer Centre in Philadelphia. The research in his laboratory is focused to elucidate the cellular and molecular biology of brain tumours, including medulloblastoma. These studies will lead to more effective strategies for patients with brain tumours. Please note that the content of this webinar represents the author's personal views and does not represent his employer in any capacity. With that, I'll hand everything over to Dr. Yang to begin the presentation. NeuroD1 dictates the differentiation of tumour cells in medulloblastoma.
Medulloblastoma is the most common malignant brain tumor in children. Aberrant activation of a hedgehog pathway is found in about 30% of human medulloblastoma. Medulloblastomas normally arise from the cerebellum, but could spread rapidly to other parts of the central nervous system. Current treatment for medulloblastoma cause loss of toxicities and improved strategies to treat medulloblastoma are still highly needed. Cerebellar granule neuron precursors, or GNPs, represent a very unique population of neural progenitors. After birth, those GNPs reside on the surface of the cerebellum. Over there, they proliferate extensively, which is supported by the hedgehog pathways. After the mitosis, those GNPs migrate inwards to start their differentiation. As I said, their proliferation is supported by the hedgehog pathways. Overactivation of the hedgehog pathways can cause medulloblastoma formations. Over 10 years ago, by conditional knockout mice, we specifically delete a patch which is the antagonizing receptor of hedgehog pathways in cerebellar GNPs. Finally, as you can see here, all the patch-deficient animals develop a huge medulloblastoma in their cerebellar, suggesting that medulloblastoma can derive from GNPs after aberrant activation of hedgehog pathways. Recently, we did a single-cell sequencing to analyze the cellular heterogeneity in patch-deficient medulloblastoma. As you can see that we got over 90% of the cells belong to tumor cells in the tumor tissues. However, among those tumor cells, there's about 20% of cells we found that they undergo differentiation. Those differentiated tumor cells express genes, highly expressed genes associated with neuronal differentiation, such as NeuroD1 and CNTN2. The TEC1 encoded by CNTN2 is a cell surface markers associated with neuronal differentiation. By immunostaining, you can see that in the tumor tissues, we found about 20 to 30% of cells were positive for TEC1. Moreover, those TEC1 positive cells, they were negative for Ki67, suggesting those differentiated cells that they already stopped dividing. As I mentioned earlier, that the TEC1 is a cell surface antigen. So by immunostaining, by an antibody specific for TEC1, we can easily isolate those differentiated tumor cells by flow cytometry. As you can see here, this is a histogram showing that the 20% of TEC1 positive cells can be isolated from the tumor tissues. We then plate those differentiated tumor cells in vitro, did a BRDO incorporation assay. As you can see here, by immunostaining, the percentage of BRDO positive cells in the differentiated tumor cells was significantly reduced compared with that among TEC1 negative cells, suggesting that the differentiated tumor cells, they were not proliferative. Moreover, we tested the tumorigenicity of those differentiated cells. As you can see here, based on the survival curve, we after transplantation with those TAG1 positive cells, all TAG1 negative cells, those TAG1 negative cells, they finally, all of the mice develop tumors with 100% of penetrance. As a comparison, no tumor was formed from the TAG1 positive cells, suggesting that those differentiated cells lost their tumorigenicities. As I mentioned earlier, that the medulloblastomas originated from cerebellar GNPs, and at the beginning, GNPs proliferated extensively on the surface of a cerebellum, or they form a specific cell layer we call the external germinal layer, or EGLs. After several cell cycles, those GNPs exit the mitosis and then migrate inward. In the deep layer, in the deep part of the EGL, or DEGL, those GNPs stop dividing and it express the early differentiation marker. As you can see here, by immunostaining, we found that the TAG1 beautifully lining in the deep EGLs, so representing the differentiated cells, differentiated GNPs at their early stage of differentiation. Similarly, by flow cytometry, we can easily isolate those TAG1 positive cells and the Ki67 positive cells, which were TAG1 negative. By RNA sequencing, we found that the both GNPs and tumor cells, after differentiation, they highly express the genes associated with the neuronal maturations. This data suggests that the tumor cells resemble cerebellar GNPs in their differentiation. And after that, we studied the mechanism underlying tumor cell differentiation. And by single cell sequencing, by analyzing the single cell sequencing data, we found that the differentiated cells highly express a transcriptional factor called NeuroD1. NeuroD1 is well established to play important roles in regulating neuronal differentiation. And after observing the upregulation of NeuroD1 in differentiated cells, we then overexpress NeuroD1 in tumor cells to examine the alterations in their differentiation or proliferation. As you can see here, that after forced expression of NeuroD1, which is the GFP positive here, you can see all the NeuroD1 overexpressed cells were negative for BRDU, and they also highly express the differentiation marker MAP2. Compared with the GFP control, the enforced expression of NeuroD1 significantly reduces the BRDU incorporation, but increase the number of MAP2 positive cells. So suggesting the NeuroD1 promote tumor cell differentiation, repress tumor cell proliferations. After that, we also intracranial transplanted those cells into SCATAC mice. As you can see here, the control, which is the GFP positive, GFP overexpressed tumor cells, finally developed tumors with 100% penetrance. However, no tumor was formed from the NeuroD1 overexpressed tumor cells. So those data suggest that indeed tumor cell differentiation is driven by NeuroD1. We then studied the epigenetic regulation of NeuroD1 expression in tumor cells. As you can see here, we performed the CHIP PCR to analyze the enrichment of H3K27 trimethylation in the promoter or enhancer region of NeuroD1 in tumor cells. As you can see here, the enrichment of H3K27 trimethylation in NeuroD1 regulatory regions was significantly increased in TAG1 positive cells compared with the TAG1 negative cells, suggesting that the neuron D1 regulatory regions is highly occupied by the H3K27 trimethylation. As we all know that EZH2 is a major component of the complex responsible for H3K27 trimethylation. By immunostaining, we found a majority of tumor cells in the tissues were positive for H3K27 trimethylation, or EZH2. However, those neuron D1 positive cells, they were mutually exclusive from those H3K27 trimethylation, or EZH2 positive cells. So all this data suggests the neuron D1 expression in tumor cells is normally repressed by the EZH2 mediated H3K27 trimethylations. And finally, we tested the efficacy of the EZH2 inhibitor in promoting tumor cell differentiation. As you can see here, this is sub-Q tumor models we generated using medulloblastoma cells after treatment with EZH2 inhibitors such as GSK126 or UNC1999. So after the treatment, you can see that the EZH2 inhibitors significantly repress the tumor growth, and the tumor size after the treatment was significantly reduced compared with the control. And by immunostaining, we further demonstrated that after the treatment with GSK126 or UNC1999, the tumor cell differentiation, which is labeled by new N, was significantly enhanced in the tumor tissues. The number of new N positive cells was dramatically increased after GSK126 or UNC1999 treatment. So those data suggest the EZH2 inhibitors can be utilized to treat medulloblastoma by promoting tumor cell differentiation. In summary, our data suggests that medulloblastoma can undergo differentiation in vivo and in vitro. Differentiated tumor cells have lost their proliferative or tumorigenic potentials. Tumor cell differentiation is controlled by neuron D1 expression, which is normally repressed by H3K27 trimethylation. EZH2 inhibitors can suppress tumor growth by upregulating neuron D1 expression. Finally, I would thank the Genomics and Molecular Genetic Group at GENEWIZ for single-cell sequencing, and the Animal Facility and the flow cytometry facility at Fox Chase Cancer Center, as well as our collaborator at Chinese Academy of Science in Beijing. I also want to thank the funding support from NCI, American Cancer Society, American Brain Tumor Association, and Cure Funding in Pennsylvania. Thank you.
Thank you, Dr. Yang, for your presentation. I now have the pleasure of introducing our second speaker, Dr. Andrea O'Hara. Andrea has over 11 years of experience in next-generation sequencing. She earned her Ph.D. from the University of North Carolina at Chapel Hill in genetics and molecular biology. She is currently a technical application specialist at GENEWIZ within the NGS team. Dr. O'Hara will be discussing the latest technology and presenting Beyond Single-Cell RNA-Seq, a roadmap to single-cell and spatial omics. Thank you, Andrea.
Thank you for that introduction, and thank you, Dr. Yang, for presenting your work using 10x Genomics to advance your research with medulloblastoma. From here, I'll be presenting Beyond Single-Cell RNA-Seq, a roadmap to single-cell and spatial omics. I'll start up by presenting to you drivers of single-cell profiling and recent developments in single-cell sequencing. From there, I'll move into spatial genomics using nanostring, geomics, digital spatial profiling, and then we'll move to discussion and question and answer. There are many limitations to the standard RNA sequencing assays. So take, if you will, a complex tissue, or in this case, a fruit salad. When we look at bulk genomics, we are basically taking that fruit salad and putting it into a blender so that we get an overall flavor profile, but we're not going to be able to tell any one particular flavor note or particular fruit. We're only going to get that overall view of what's happening. Single-cell genomics is able to take this to the next level so that we can identify all of the individual fruits and how much of them they're present, so that we're going to get a better resolution of what's occurring within that complex tissue or fruit salad. Now, with any research project, the quality of our input directly dictates the quality of our output. And so with 10x genomics or single-cell profiling, we really want to focus on having lots of live cells for our downstream analysis. Dead cell removal improves the data quality. At GENEWIZ, we have a proprietary protocol where we're going to be using dead cell removal using magnetic beads. So for any sample that is below 70% viability, we will do dead cell removal and use these magnetic beads to remove the dead cells so that the live cells are left. With this approach, we will have increased cell viability, and that ultimately yields the best possible result, with improved quality in both the sequencing itself and the number of genes that we can capture per cell. Now, when we're thinking of single-cell sequencing, we want to move beyond just single-cell RNA sequencing. So at GENEWIZ, we have a comprehensive suite of single-cell sequencing solutions. This includes single-cell RNA-Seq, but also includes single-cell ATAC-Seq and single-cell immunoprofiling. Single-cell ATAC-Seq allows for epigenomic insights at the cellular level. This includes, but is not limited to, cell cluster and classification to help identify cell types in heterogeneous cell populations, regulatory variation to detect altered gene regulation and/or novel enhancers in rare cells and subpopulations, trajectory studies to discover states for lineage and development tracing, and that way we can understand the mechanisms of cell fate. And then finally, chromatin accessibility. So by using this ATAC-Seq approach, we're able to reveal subpopulation-specific regulation, which allows us to find cell type-specific cis-regulatory networks. Single-cell immunoprofiling really enhances the overall view of our immunoprofiling studies. Standard immunoprofiling is a bulk analysis where we're going to take all of the cells that we analyze and look at the VDJ, B cell receptor and T cell receptor and get an aggregate or overview of what's happening, which means that we'll only have the heavy chain and the light chain. We'll be able to tell the frequencies of heavy chain and frequency of light chain, but we will not be able to tell the complete pairing of heavy and light chain. With single-cell immunoprofiling, we'll take all of the cells that we are looking at and we'll get the complete heavy and light chain combination. So we'll be able to actually get a comprehensive view of exactly what's being expressed in all of the cells assayed. It's a much stronger and more comprehensive view of the exact immunoprofile of the sample in question. Using these all together, we can get a great multi-omic characterization using these single-cell platforms. So we can use the single-cell RNA-Seq to characterize rare cell types and complex systems such as cancer, perform high-throughput genetic screening, and identify novel biomarkers. We can use the single-cell ATAC-Seq in tandem to uncover epigenetic variability, define cell types and states for lineage and developmental tracing, and discover novel biomarkers as well. And finally, integrating the single-cell immunoprofiling will allow us to investigate both clonality and diversity and also assemble and annotate entire VDJ gene sequences. Now, again, all of these do require us to have high cell viability because what we put in upstream dictates what we can get downstream. So in addition to dead cell removal, we highly recommend our proprietary cryopreservation protocol. This freezing protocol allows for excellent recovery after acceptance of samples so that we have high cell viability and then can move forward for future studies. And so I'm just showing you here on the bottom right the difference between fresh and cryopreserved samples. You'll see that the cell viability is on par for fresh and cryopreserved. So really, really critical that not only do we have lots of good, healthy cells, but that they are well preserved in order to attain that. Now, once we have these great healthy cells that we have frozen, shipped appropriately, received, and then done dead cell removal as needed, We can do excellent multi-omic characterization. So here I'm showing you an example of a thymus. With single-cell genomics, we did single-cell RNA-Seq shown on the left and single-cell ATAC-Seq shown on the right. And what we're able to do is actually overlay this information together to determine both the regulation pattern at the DNA level of what can be expressed, and then the expression pattern on the left showing what actually is expressed. So we can combine together regulation component with the actual expression to see a comprehensive view of what's happening in the entire cell. Now, this approach is actually using two different cell sampling from the exact same tissue. So we're using some for single-cell RNA-Seq and some for ATAC-Seq. Again, we're still looking at the same sampling, but we're using slightly different cells. Advances in single-cell profiling are introducing the 10x multi-omic assay. So you can see here on the left, I'm showing you the example I just described where we are showing a split sample. So some cells are going for gene expression, some are going for ATAC-Seq, representing the same whole population and theoretically representing the same number and types of cells. But the new 10x multi-omic assay will allow for simultaneous detection for ATAC-Seq at the DNA level and gene expression on the RNA level. So we'll be able to get a complete comprehensive view of each individual cell together, allowing for just seamless analysis. This will be the gold standard, very cutting edge moving forward, because it will allow us for a comprehensive view of exactly what's happening in every cell that's analyzed. Now, of course, there are limitations to single-cell assays. So when we think of our complex tissue or our fruit salad and we go to our single-cell genomics, we're going to be able to tell all of the different kinds of fruit that it's made up of and how much of each particular fruit, but we won't be able to tell where those fruits are in the fruit salad. So we won't be able to know which cells are by which cells when we're thinking of this in this spatial tissue form. And that's where spatial genomics can really take this all to the next level. So if we think of bulk RNA-Seq, we're going to get a high-level overview, but we're not going to capture any heterogeneity. We're really getting one uniform view of that entire tissue as a whole. This is really great if I have something that's going to be very homogeneous, but if it's very heterogeneous, I'm not really going to know what's making up what. Single-cell RNA-Seq can really help with that because we're able to characterize the heterogeneity of the population, but we're losing all of the spatial relationship. And that's where spatial genomics can really come into play. We're going to be able to use that tissue specimen itself for the RNA or protein expression, and we'll be able to maintain that, that cellular context while also doing the assay. So what does this look like? Here's a good example. So if we think of spatial genomics, we can take, for example, infiltrating tumor. You can imagine now here we have on the left areas that have a tumor that are indicated by our marking, as well as the normal tissue. And on right, we have the same. Now, if we looked at these together with our bulk approach, we would get the same amount, basically, of tumor versus normal. If we did this with single cell, we would get similar information, and we would know how many of the tumor cells and how many normal cells and how they interacted with each other, but we would miss all the spatial context to know that if we were looking at these two tissues with spatial genomics, we'd be able to very clearly see that they in fact have far different cellularity, and we can see where these cells are interacting with one another and how they might vary from one another when we look at the final end result. So what that means is that bulk RNA-Seq, while being fast and most cost-effective, is really ideal for homogeneous samples only. We'll get transcriptome-wide quantification, but we lose all heterogeneity. The single-cell RNA-Seq option allows us to profile thousands of cells with single-cell resolution, which is good for capturing heterogeneity, doing combined gene expression with a VDJ repertoire profiling, and including DNA information with ATAC-Seq, but we lose all cellular context. So we'll get a good idea of what all of those cells look like, but we won't know what's adjacent to where. Nanostring geomics digital spatial profiling provides morphological context for simultaneous profiling of protein and mRNA expression. We're able to select large regions of interest or single cells. We have a lot of flexibility as to what we choose, but we may be slightly more limited into how much information we can get. We're going to be using curated panels or process-specific disease panels that can include tens or thousands of transcripts, but not the entire transcriptome.
Thank you, Andrea. That concludes our presentation for today. We hope you enjoyed it, and thank you for joining us.